My research is involved in a condition called familial hypercholesterolemia, and this is an inherited high cholesterol condition. And basically, these people have a mutation in a protein that takes the bad cholesterol out of the blood. So they have far too much bad cholesterol, and they start developing what we call atherosclerosis, which is hardening of the arteries. They generally develop high cholesterol sort of in their late 40s, and they generally kind of present with a heart attack in about their 50s. Familial hypercholesterolemia affects 120,000 people in the UK, which is equivalent to things like Parkinson's disease. That's about a similar incidence. So I work on gene therapy, and gene therapy is looking at how we can use the gene as a drug. So giving people who don't have a working copy of the receptor, giving them a working copy of it. So basically, the working copy then starts to remove the bad cholesterol from the blood, and that's called gene replacement gene therapy, which is what I'm trying to achieve in my animal models and cell models. There are thousands of alternatives to doing animal research, and before anyone can even think about doing anything in an animal, you have to have completely exhausted every possible alternative. So, 95% of my work is looking at cells in a tissue culture dish, or taking protein out of cells, doing everything I possibly can to understand whether it's going to work in an animal before I even think about going and doing it in an animal. When I first started, we'd designed this type of vehicle that we thought was going to work very well. It pretty much cured the condition in cells in a dish. And then when we went into animals, it did absolutely nothing. And that could not possibly have been predicted from our tissue culture experiments. And so we then had to go back to the drawing board. And I actually think doing the animal experiments, finding it didn't work, and going back to the drawing board has left us with a much better tool, and a tool that's going to work in a much more intelligent way than what we had at the start. So our animals are kept in a very clean area of the lab. I have to put on full scrubs, cover my hair, wash my hands with ethanol, wear gloves all the time. Some people even wear masks if they're concerned about any kind of bugs they might have, and wear overshoes. And that's pretty much what we do to keep them clean. The other thing is that the animals have their own air supply. And then when we get the animals out, we actually put them into hoods where they are completely protected from us and the environment around them with clean air. Because what we do is work under a license from a government body, everything we do has to be called a procedure. So in our work, a procedure is a genetically modified animal. So my animals being born, that's a procedure. Sometimes they have a biopsy of their ear done, which is pretty much like a human getting their ears pierced. That's a procedure. Taking a blood sample is a procedure. But most of the procedures that we do are just animals that we have been born, and that's pretty much it. So in my research, we use quite a cool refinement, which is to actually use a camera to see gene expression in living animals. And this means instead of having to have a lot of animals that I've done an experiment to and having to sacrifice them at time points that I need to understand gene expression, I can just take an animal and look at it in a camera. It's very non-invasive, the animals don't have to be harmed in the process, and we can understand gene expression without having to use so many animals and without having to do so many procedures to them. The new Biomedical Sciences building has made my life a little bit harder. It's a bit of a hassle to have to get over there to be clean all the time, but for my mice it's made a difference. They are happy, they're healthy, they're looked after by an excellent team of technicians. So my experiments are going very well because my mice are happy and healthy, which makes me happy. In terms of getting a gene therapy cure for FH, I think we're probably 10 to 15 years away from that. But I think that the progress that I've made in the last couple of years may have brought that a little bit forward, and I am hopeful that sometime in the future we will have a gene replacement gene therapy for FH.